Okay. So we'll continue now on sort of the rest of the slides, or the actual sort of beginning of the real material for this course. Um, and so these slides should go fairly quickly because they're just kind of a review. But feel free to look at them in more detail, or if I move too quickly, just ask questions when you feel uncomfortable. So to kind of start off with really, really basic definitions, but ones that I think are quite important to at least make sure everyone's on sort of the same page. And that's sort of what is an image. And so a lot of you will probably have very different definitions of what an image is and what it sort of means. But the very abstract definition that we're going to go to is a pairing between spatial information, sort of position, and some other kind of information, value. And so this is sort of a very abstract definition. Most of you would think, what you take with your phone is an image, and it is, but it's a lot broader than that. And so we have this definition as just between spatial and other information, because, if you're looking at confocal microscopy images, that relationship is very different than if you're looking at micro CT images. And both of them would still be considered images, but we know they come from very different sources. They're measuring very different things and have very different meaning. And so in most cases, this is a two-dimensional position, so kind of an x, y coordinate, and a numeric value, so sort of an intensity, or in the case of color images, a color. And so, if we have a basic image and a sort of simple representation, I don't know why all these are so small, we have an image that, maybe 5 by 5, and we have this pairing of x, y, and intensity. And so we can see, this table view of an image, which is the simplest representation we have, but obviously not very helpful for seeing what's actually inside the image. And then for all the students, this code, don't feel overwhelmed by it or confused. It's really just to show you where everything comes from. And so if you're interested, you can look into more detail. And if you're not, just completely ignore it. But don't let it sort of confuse or overwhelm you. It's just there to have an idea of where all these charts come from. Because at least when I was taking courses, I found it quite frustrating that you would quite frequently have graphs or images or figures shown, and you had no idea where they came from, or they would sort of skip over 5 steps in order to get there. And I found that if you always show exactly where everything comes from, even for the simple things, it makes it a little bit easier for some students to follow. But if you find it overwhelming, just ignore it. And so here is where we now have kind of the more standard representation of the image, where you see, the image as sort of this square. We have all the different intensities, and then we have some sort of color mapping the intensity value to a color between black and, or dark green and yellow. And so, it's just a noisy pattern. There's nothing actually significant in there. But this is the standard way of looking at it. Most of the time, you don't look at an image as a table. And so when you have 2D intensity images, kind of the first thing you do is apply a color map. It's also called sort of a lookup table or an LUT, so that the image is sort of a bit more exciting and a bit easier to interpret. And so particularly for those of you who do fluorescence or confocal imaging, you might see these pretty pictures of cells where they're, bright green, but the cell isn't in fact bright green. That's just the lookup table they've applied to the image in order to make it look that way. And so here we see that we could also use blue, for example, as the lookup table, where we can now show each one of the values in the image and map it between white and dark blue. And so that gives us a slightly different representation of what we're doing. And we can arbitrarily define this lookup table to be whatever we want to visualize or point out in the image. And so this is one of the areas you very quickly get into trouble in science, is that if you change the lookup table, you can very easily change what you see in the image. And so if you're trying to show, look, we have this drug that makes cells much healthier, and I use one lookup table for the healthy cell and one lookup table, or for the sick cell, and one lookup table for the cell after my treatment, then you can end up showing a major difference, even though there is almost no difference, because the lookup tables make you interpret it differently. And so here, we use JET. This is the default lookup table in MATLAB. You're strongly encouraged not to use this lookup table because it's not well suited for sort of the visual perception system that we perceive sort of yellow and red much more intensely than we perceive orange and green. And so it causes you to have a skewed representation of what's actually in the image. And that's where this blues and other color scale that we had before, this is called Viridius, are much better suited for the image visual system so that your eyes don't overestimate the differences that are actually present. Another lookup table for the same data is HOT. And if you were to look at, these two images, you would potentially come up to different conclusions because of what your eye starts to see with these different tables. And so basically a lookup table is just a function which takes this intensity and spits out a color. And so, we can have these three different lookup tables. Yeah. Yeah, that's a lookup table as well. But it's also not that easy because, black-white gets displayed on your screen, which is displaying everything in color. And some screens have darker blacks and brighter whites than other screens. And so you always have these issues of making sure your perception of your image is the same. And so there's not like one perfect lookup table for every problem. It's just something to be kind of aware of, that when you're using images and processing them, that the choice of the lookup table will have a pretty significant effect on what you're looking at. And so here, if we just look at the red component of the different maps, so that each one of these maps shows the intensity on the bottom and how much red is present in the image versus, as you increase the intensity. And that we see these trends kind of are very, very different where, the white to blue goes from high red to low red, but it's also not a kind of linear curve. It's a very shifting curve. And, the jet one is flat and then becomes very steep and then gets flat again. And so you have all of these sort of simple linear responses that you have to be aware of when you're doing these tables. You can also do sort of more complicated transformations here. So one of the common things to do is to do sort of a logarithmic relationship between the intensity and the color that you use. And so here, instead of having sort of this hot intensity go kind of linearly and up, you can actually have it be logarithmic. So if you were visualizing something, in seismic imaging, for example, you would want to have a logarithmic relationship between the intensity of the earthquake and how you would plot the color, because you don't expect linear things would blow out quite quickly and not give you a reasonable visual of what's going on. And so we can take here the same image where we have grayscale, the HOT, and the grayscale log. And that if you were to look at all three of these images, you would come to very different conclusions about them. So in this image, I would say the holes look the most pronounced. So if you were to ask which bone is most likely to break, you would probably, or I would at least say this one. This one looks like it's the most filled in. But of course, these are exactly the same image. It's just different lookup tables that are put on it. And so it's important that you're kind of aware that a lot of the things that you visually perceive aren't necessarily in the data. It's in how you make a representation of the data. And so just to know, trying a bunch of different color schemes, switching out the panel can help you be more aware of what your eyes are potentially overemphasizing, because the difference between black and yellow, your eyes perceive more strongly than the difference between black and white. So now to kind of go on to other types of images, that was 2D images with just intensity. For 3D images, you just have one more dimension in space. And so rather than just having an X and a Y position, you now have a Z position, or T if it's a movie. And so we end up with this kind of same sort of representation where, instead of having a 3x3 image, we now have a 3x3x3 image with all the values in it. And they can be sort of rearranged in a table form, or what we commonly do in this class is make sort of a montage out of it. And so that's where you take each tile and sort of place them next to each other so that you can see the whole view as a bunch of 2D slices, because it's a lot easier to interpret a bunch of 2D slices than it is to interpret a 3D image for most things that we're looking at. Then we kind of get to the last thing, which is where you have multiple values. We have the position dimension and the values dimension. And so, if you have more position, you go to 3D, 4D, 5D imaging. And as you have multiple values, you just get sort of a greater depth of what you're looking at. The intensity images are what we started with, but color images are kind of the most obvious application of multiple values, where you have sort of red, green, and blue at each pixel. And so each pixel has a red, green, and blue value that you show. Many types of images even have a transparency channel. And so if you work with like PNG images very often, one of the channels is transparency, which allows you to sort of overlay images quite well so that the background doesn't cover anything up. And, for clarity, we call this the dimensionality is where the spatial position comes in. And so if you have a 3D image, you're talking about having 3 spatial dimensions. And the depth is the number in the value. And so, you wouldn't call a color image with X and Y position a 3D image, even though you do have 3 coordinates there, because 3D is referring to just the spatial, and the dimension, the depth would be then 3 because you have red, green, and blue. And so here we sort of have an example where you can see that you have this sort of image that has intensity and transparency. You might get measurements like this from a certain device where you can then start to show them and say, we have one intensity image and one transparency image. So at each point, we know what the intensity is and we know what the transparency is. We could then also show them on sort of the same axis where we have, the intensity shown as a color and the transparency shown as a size. And so that we can start to visualize those two things together, but how we combine them is kind of completely up to us. So there's unlimited flexibility for how you want to show these things. It's just kind of an awareness that there's not one correct representation, and that you should probably look at a few of them to get a better feeling for your data and not be sort of tricked by your eyes too easily. This then extends to hyperspectral imaging. And did anyone have any hyperspectral imaging applications? Has anyone heard of hyperspectral imaging before? No one. Okay, so hyperspectral imaging is actually quite interesting. So it's very common in satellite images. So when you're looking at like agriculture, and so rather than focusing on just, red, green, and blue like a normal camera, that you focus on a number of different wavelengths. And so in particular, this is helpful because you can look at things like different infrared wavelengths where you see heat coming off, or different very narrow bands in green where you can identify chlorophyll, which allows you to actually use satellite images to determine what time is best to sort of harvest crops. And so you can get satellite images of the whole Earth. I think the newest companies now deliver it sort of on a weekly basis. And then you can use that to actually make decisions about what, how much fertilizer you use and when you harvest crops. And then obviously there's the whole spectrum of military applications that when you can identify very narrow wavelengths, you can differentiate very specific types of materials. From the medical area, there's a number of different techniques that use this. So IR spectrum are people familiar with? I mean, so in organic chemistry, some of you might have gone over it, where you take substances and you do a spectra on them to see what molecules are present. And so this is a very similar application called Raman spectroscopy or Raman imaging, where basically you take a spectra at each point in your image. And so here you go through the image and scan it line by line, and at each point you take an entire spectra of information. And so kind of what that gives is we have an x coordinate, a y coordinate, a wave number coordinate, and then a value. And so it's actually a huge amount of information to kind of process because you have all of this different information together. And how do you visualize that sort of 3D mess of data? And here you can kind of show that, you could group it by x and y and sort of show what the average or what all the spectra look like inside there, but it's not particularly insightful. And we'll need to come up with better techniques for visualizing this data well and starting to simplify it so that you can actually come up with, how healthy are these cells, what's the presence of protein and DNA inside these cells. So to kind of move on to the next aspect, this is just imaging in general and sort of how you understand imaging systems. So I think it's also probably mostly review for a lot of you, but trying to put everything into kind of the same framework. And so with all imaging systems that we deal with, there's some kind of impulse or excitation. There's then a characteristic of the sample that we're looking at, a response that comes off the sample, and a detection system. And so, the impulses, in the case of your, iPhone would be the light in the room. So you have lights coming out that are shining on objects here. The characteristics, in the case of light would be sort of the electron shell levels of absorbing and reflecting light or the optical properties of the sample that you're looking at. In other cases, they might be phonon energy levels if you're looking at sort of infrared spectroscopy, or electron spins or molecular mobility. And your response can then be, is it absorbed or not? Or how much of it is absorbed? Is it being reflected? Is there a phase shift? Is there some sort of scattering? Is there some kind of emission? And, classifying it into one or multiple of those groups. And then finally, you have the detectors, which can be your eye, light-sensitive film, a CCD or CMOS camera in the case of most sort of standard imaging systems, a scintillator, so if you're using X-rays, you typically aren't able to detect X-rays directly, so you have a small plate that the X-rays go onto, and then you're able to detect whatever comes off, or for ultrasound, a transducer. And so that all of these are all imaging systems, they all work in different ways, but they have the same fundamental setup. And so that we have, again, way too small here, the sort of, a number of different examples where, light microscopy is sort of standard. It's the same basic principle as how your eye works. You have white light, electronic interactions, absorption, and your response is sort of a film or a camera, all the way to kind of more extreme cases where, atomic force microscopy, you have a very sharp tip that you're dragging across the surface, and when it hits something, it moves, and you measure that sort of deflection of a tiny mirror. And so you're measuring just sort of small changes across the surface rather than measuring, electronic interactions or molecular mobility or some of these other factors. And so this isn't important to know. It's just more important to be aware of all of these imaging systems rely on the same basic setup. So when we talk about acquiring images, there are other classes that get into much more detail for this. What we'll talk about is just insofar as it's important for the image processing or image analysis steps. So what most of you are probably familiar with are sort of traditional or direct imaging approaches, where visible images produced can easily be made visible. And so where you're capturing sort of direct image data in what you're looking at. And so this would be optical imaging. This would also be microscopy, where you get sort of images out of what you're looking at. And so here we have this kind of simple example where you have your original object, you have your measurement, and then you have your reconstruction. And so maybe when you're taking a measurement here, you know, the object gets blurred out, there's some kind of defocus, there's some sort of, you know, flipping going on, but fundamentally you get something that resembles your original object fairly well, and that you're able to then do a reconstruction to start to get closer to what the original object looks like. Obviously, there'll be some noise and things that are added that you won't be able to get rid of. The opposite, or the sort of counter of this, is this idea of indirect or computational imaging, where the recorded information doesn't really resemble your image at all. And so this is where you need to transform what you collect in order to produce an image. And so where this comes in is where you have sort of diffraction patterns. So in this case with sort of X-ray diffraction, where you measure something that looks like this, so it has no connection or no resemblance at all to what you were trying to image. And then you have your kind of original object. And so you need to do some kind of reconstruction on this diffraction pattern in order to get to your original image. And so those are the two primary pathways you have for getting images, depending on what kind of system you're using. And so, you know, MRI, diffraction, some ultrasound systems work like this, where you're not able to capture an image directly. You capture lots of other data that you can then use to reconstruct the image. And so here is sort of the traditional imaging, or sort of the direct imaging approach, where you have some sort of light source, an object, a lens, and then you have your captured image as your output. And the traditional image model. And so this is math you really don't need to worry about at all, but that basically for the most standard image problems, you have a light source, which has some kind of profile associated with it. You have your object, you then have this sort of free space propagation, and so when light travels in free space, it sort of blurs. And then your whole microscope optics, which can kind of additionally blur or add artifacts to your image. And then finally your detector, which has sort of noise associated with it, and then some sort of dark current, because detectors are always reporting something even when nothing's coming on them. And so you have to have a system where, you know, you account for all of these different factors combined together. And then when you're dealing with color images or multiple different wavelengths, you then integrate that over all the possible wavelengths that could come in. And so basically, you know, as for the third lecture, we'll kind of get into how you can get rid of or eliminate as many of these terms as possible. And so in reality, you know, we can write this nice equation, but we don't actually know exactly what that looks like. We usually don't really know what the beam profile looks like. We're not actually sure how far away it is. You know, all of these things can change with time and temperature. And so even though we can write this nice equation, it doesn't necessarily mean we can solve it. But it's trying to say like how our image gets destroyed before it gets to our detector, so that we know what we should be trying to reconstruct, and to appreciate that the things we measure are often very disconnected to what the actual sample looks like. So through computational imaging, we sort of have the same sort of problems where, you know, with tomography, you know, you're rotating a sample and you're able to get a 3D image by taking a number of 2D images and then reconstructing them into the whole 3D objects. Micro lenses, so for like light field photography, you have like arrays of micro lenses where you capture different angles at different positions on your sensor. Where that doesn't give you an image, you have to take all that information, put it together in order to extract one. Diffraction patterns, as we sort of showed before, hyperspectral imaging, surface topography. So all of those examples where you're not actually measuring the image directly, you're measuring data that you then have to use to reconstruct the image from what you're looking at. So now to kind of get to the more broad overview of the course, so for some of the ideas of image analysis, I've broken it down here into kind of 3 different approaches that we take. And so right now in the course, we've said an image is a bucket, more or less, of pixels. So you have all of these different positions and values put together. And the question becomes, how do you analyze this easily? And really, how you take information out of these images depends a lot on what approach you have for these problems. And so the first approach that we have is this experimentalist approach. And so that's kind of most of you not coming from a computer science background probably have this approach where, you're really problem driven. It's top down and it's reality model based. And so you're given an image and you know that there should be cells inside it and you want to count how many cells are there. And so, you have some model of the world, you have some expectations, and your goal is to try to extract that in a reasonable way. The second, for maybe more of the computer science or software engineering approach, is this sort of computer vision. And this is where you're method driven. You sort of have feature based and image model based. And so for all of you who've done things with sort of, filters and edge detection and feature detection and matching and all of those, that's really the computer vision approach. And so how can you apply these techniques to the images you get in order to quickly extract the information you're looking for? And so you're not actually, trying to find cells or count cells, you're trying to find features, and then you'll take all the features together, you'll maybe match them across a number of images, and then at some point maybe you'll get to cells, but the approach is really driven by, what mathematical tools are available that you can apply to images in order to get a result. And so, face detection also works this way, where you basically go through the images and look for certain patterns that look like faces, and then you quickly apply this to lots of different images in order to extract faces from it. The last approach is sort of this deep learning approach. And so this is much more results driven and biology inspired. This could also be the more sort of machine learning approach, if you wish. And it's where you build both sort of the image processing and the analysis from scratch. And so these are kind of the sort of tasks where, the model-based approach and the computer vision-based approach would clearly fail. So if you look at something like, captioning images, I guess most of you seen that demos before with Google? I think it's, show and tell, where, it's able to take this image and it's able to respond a person on a beach flying a kite. And it's able to take, sort of other images, 2 brown bears sitting on top of rocks, where, if you've done some programming or some image analysis, this should kind of blow you away, because trying to come up with a model that's able to take this image and figure out that there's a person, a kite, they're on a beach, is unbelievably difficult. Maybe identifying that there's a person is a problem you could solve, but taking a random image, finding a person, seeing the kite, making the association between the person and the kite is a hugely complicated problem, particularly when there's thousands of different things it could be. A blue and yellow train traveling down train tracks. I mean, it's another, you could probably say there's blue and yellow in the image, but more than that would be really difficult. And so this is where, you have approaches that you use to solve problems that are very, very difficult, where you couldn't even begin trying to describe a list of rules you would use to actually quantify what's in this image. And so that's, obviously one of the more popular approaches being used today, and things that we'll touch on a little bit. But our focus with this class will really be about looking at the different approaches and how to choose which one makes the most sense for the problem that you're trying to solve. So kind of on science quickly, we'll try to wrap this up fairly quickly. But basically, the point of doing science, the point of doing all these experiments is to sort of discover and validate new knowledge. We want to use the scientific method as an approach to convince other people, so that the reason you follow the scientific method of forming a hypothesis, testing your collecting data, testing your hypothesis, and then making a decision on this isn't for yourself. It's so that you can convince someone else that the things you've done are meaningful and are reasonable basis for sort of making new experiments and making new hypotheses and testing them. And so the point for this is that, while qualitative assessments are quite important, you want to have results that anyone can reproduce anywhere. So the point of science is that if you do an experiment here, someone else doing that experiment in China or Antarctica should get exactly the same results if they follow the same steps that you performed. And with imaging, this sort of gets more complicated because they're great for qualitative things. You can see in images very quickly, are there cells? Do I see a crack? Is this material connected? But it's quite difficult to interpret that without large programming investments. That there's, for a temperature, it's a number. It's 50 degrees Kelvin. You can measure it 5 times and then average it, and that makes sense. If an image, you can take 5 different images, but if you average them together, you now have a blurry image. And it doesn't necessarily help you understand how noisy is the process or how reliable is the tool that you're using. It's then kind of further complicated by the fact that there's so many tools available. So if you read through papers today, you'll find people using MATLAB, people using ImageJ, Fiji, PyTorch, TensorFlow, thousands of different tools available, ITK, and everyone does sort of different processes. And so it's a bit overwhelming of how am I supposed to copy a process that requires me to install software for 3 months on my computer and use all these different components with exactly these versions in order to get the same result. And so that's kind of the basis for why we're doing this, is how can you establish sort of a simple, easy way that you can give to someone else and they can get exactly the same results as you. The then last thing we'll go over is this sort of idea of quantitative imaging. So human eyes have major problems, as we sort of started with this LUT problem, or lookup table. And if you ask which center square seems brighter, which one? Yeah, so the one on the left looks a lot brighter, but if you look here, they're exactly the same value. And so if you were, for this problem, it's fine because we actually don't really care which one it is. But if this were, a tumor in your lungs and your doctor decided not to treat it here because it doesn't look that bright, and here because it looks very bright, you would be presumably quite upset that your treatment was kind of arbitrarily being decided by the fact that their vision system wasn't very accurate and not based on real quantitative criteria. And so there's a lot of things that human eyes are really good at, but there's a lot of things they're also very bad at. And so what we want to do is make sure we're quantifying things as much as possible so that we don't end up with too many drawbacks because of the things the eyes are bad at. Similar sort of things kind of are the intensities constant across the stripes. We also have the problem that this is then being projected and scaled and everything else, but is each stripe of constant intensity? And, it isn't. They look like they're sort of darker on the right edge and brighter on the left edge. And of course, they are of constant intensity. You can go through and check. They have exactly the same intensity at each point, but because of the way our eyes visualize that, when they see a bright color next to a dark color, they do some sort of automatic edge enhancement for us, which is helpful if you're trying to make out a cheetah in the wild, but it's very unhelpful if you're trying to quantify an image. And so our visual system really isn't built for handling images where we're trying to say sort of absolute quantitative assessments of something. And so science demands sort of repeatability and really wants reproducibility. And so, what we want to do with this class is keep track of what we're doing so that we have everything sort of organized in an easy to understand packaged way. And I think we'll get into sort of these recipe examples next week, where we cover that and then the ground truth basis for this. But basically the exercises, or if you're interested in doing the exercises, what they'll cover today is sort of how to use this tool KNIME. So we won't do any Python this week, and how to get it installed and set up on your machine. So for everyone in the class, we have enough accounts for everyone to use these computers. But if you have your own laptop, it's probably better to do it on your laptop, because you can then take it with you. The accounts on these computers, I think, are deleted 3 months after the semester ends. And so anything you've saved will be gone. If you're in electrical engineering or BME, you should have access to these computers through your normal ETH account, so you can do things there. But I think for most of you, using your laptops or your own machines is probably a better way to start out, because then you can continue doing your analysis after the course ends and don't have to move everything over. But if you want to use the machines, you're more than welcome to. And that this kind of shows how to at least get started with KNIME and open up a few of the kind of basic, how do you load an image, how do you look at it, and how does KNIME work with what you're doing. And so I think I had a slide on that opened. So if you Google KNIME, it's sort of an open source free tool where you can combine lots of different analyses together. And so here, there's a company behind it. The company's in Constance and Zurich, so they're actually not very far away, and they make most of their money selling consulting services and selling like server licenses for it. But for using, it's free to use, free to install anywhere you want, and open source, so you can make any additions or changes you want to it. So it's not sort of completely free and open like other tools, but fairly close. And I think what makes KNIME quite useful is that you have these workflows that you make where you keep all of your steps very distinct. And so here, this is not image analysis. This is just sort of a normal data processing where you kind of load in data, you combine it, you do some transformations, you then kind of fit, decision trees or models to it. You then make predictions. You can then visualize your results, put your results in a database, export models, do kind of all of those steps in one tool. And one of the things that makes it particularly useful is that you have integrations to kind of all the tools you normally use. So you can have KNIME call a MATLAB script or run Python code or run sort of bash or shell scripts, run R visualizations. And so you have all of those things available quite easily. And so I think it makes it a lot easier to get started and for other people to understand what you've done. And so if you look at a giant Python script someone's written, it's very difficult to figure out what they're doing, where the parameters are, what decisions they made. And if you look at something like this, you might not understand exactly what's going on, but you can quite easily, click on this decision tree learner and go to properties and see what settings they set, how many trees they use, or how they trained the tree, how they pruned the branches, and kind of tweak all of those outputs. And so here, it makes a lot of that a lot more accessible to people who aren't programming. And most of your advisors probably aren't going to be as good at technical things or programming as you are. And so if you need to communicate or have discussions with them, having something like this is a quite easy way to show them what you did, rather than having, big giant piles of code that they can't do anything with. And so, you won't need it. So all the exercises you can either do sort of in KNIME or Python, but KNIME is a fairly useful tool to learn. And if you go through here, there's thousands of nodes that are available. So, you have image processing tools. It's also quite nice because, if you go to filters, you see all the different types of filters that they have. And so you can click on one of them. You can read a description about what it does. You have all the different parameters that you can tweak for it. And you can, replace it with another kind of filter and run it again and see what happens. It also makes it very easy. So if you're dealing with sort of a workflow, you can right-click on a node, go to execute. And if you wanted to see what the output of this node was, you can just right-click and view it and see kind of what all the intermediate steps were for your analysis to get done. And so I think particularly when you're learning, it's very helpful to be able to kind of do that debugging and testing interactively and sort of see how different filters or different results look with kind of immediate feedback. And so, yeah, so the exercises will be more just getting comfortable with this tool, because even for the people who program Python, it's not a bad tool to be familiar with, and it helps you kind of understand these workflow concepts a little bit better. And so, are there any questions? Okay, so I'll let you work, and if you run into issues with any of this, just let me know. If you want to install KNIME on your own machine, I'm updating the slides at the moment, but if you just go to, so with the course website, if you click on View on GitHub, has anyone heard of GitHub before? A number of people. Who hasn't heard of GitHub? No one? Oh, a few people. Okay, one or two. So GitHub is basically a place where you can store code, projects, and other things, and it keeps track of all the versions. And so you can see that, for the course website, there's been 18 versions. Who's made them? These X's mean they didn't pass all the tests, so I put code up there that was buggy or had problems with it. And it has issues and pull requests. And so what are quite nice about issues is if you have a problem, you can just make a new issue and list it up there. And here you can, for example, click on the Add Updated KNIME Links and then see for Windows and Mac, you have this sort of prepackaged KNIME that you can download. You can also set it up yourself by going to the KNIME website and installing, but this has everything set up with a few workflows that run and all the packages you need. And so it makes it a little bit easier to get started because you don't need to install all the packages first. Because if you follow the normal instructions, you'll need to make sure you have sort of the KNIME image processing toolkits installed with the latest versions, and that can take a little while to get set up. But yeah, so you can start working.